My name is Nadia Terrazzini, and in this short presentation I will tell you about my latest project made possible by funding recently and gratefully received by the Centre. In this project I will design a new assay for the detection and monitoring of immunity to SARS-CoV-2, and the new assay is a FLISA, which stands for Fluorescence Linked Immunosorbent Assay. I will carry out the work with Cherry Collins and from the Centre, and the work will be possible also by collaboration with BSMS, where Professor Florian Kern will provide plasma from COVID convalescent patients, and I also have set up a collaboration with Michael Cave, an ex-University of Brighton student who is now director of a small Cambridge-based company called Brighton Diagnostic. In the current COVID-19 pandemic, a huge global effort has been made to develop sensitive tests to assess immunity after infection by the SARS-CoV-2 virus. Sensitive assays are particularly important at the current stage of the pandemic, as they will allow us to monitor the antiviral immunity raised not just as a consequence of infection but also after vaccinations which have now been distributed in international immunization program. A sensitive, reliable antibody test will allow us to identify those with immunity for COVID and indeed show the true infection rate within a population. Sensitive antibody tests would let us understand more on the dynamic of antibody response. For example, understand antibody levels within different disease severity cases. They will also support the monitoring of immunity in immunized recipient and let us study the duration and persistence of antibody levels after vaccination. ELISA is a well-established technique for the detection of antibody, and indeed a number of ELISA kits have been already validated for the serological detection of antibody against SARS-CoV-2. This project will optimize a novel assay format based on the fluorescent detection of antibody. So the new assay will be a fluorescence linked immunosorbent assay, FLISA, based on the use of CPN, conjugated polymer nanoparticle, which will be provided by Brighton Diagnostic. The new fluorescent-based assay will have increased sensitivity, reduced background and assay timing, but will have similar test costs to traditional commercial format. So, similar to an ELISA, recombinant SARS-CoV-2 spike protein, the S1 subunit, will be coated on wells target for the specific antibody in the plasma. But different than an ELISA, in the FLISA, biotin-related antibodies, so for example here anti-IgG antibody, are bound to streptavidin C and P, which will be fluorescence and magnetic. Hence, unbound CMP antibody can be eliminated applying a magnet to the inverted wells. And fluorescence can be measured on a fluorescent plate reader to calculate the amount of antibodies in plasma. Antibody titers detected with this new ultrasensitive FLISA will be compared to the gold standard to a gold standard commercial ELISA kit. We will test samples from plasma from non-infected and COVID-19 convalescent patients recruited at BSMS Medical School. And to avoid waste of COVID convalescent plasma sample, initial optimization of the assay will be performed for the detection of IgG antibody against the tetanus toxin antigen in normal plasma, for which we are all being vaccinated.